Alright, where we left off the last video, we had just taken a bunch of bacteria and inserted some human genes into them, so some restriction fragments, and so we would have had a plate with a whole lot of bacteria growing, and we would be able to figure out which ones had human DNA in them. But if we're doing study on a specific human gene, then we have to sort through all of those bacterial colonies and figure out which one or which ones have the gene that we're looking for in them. So there's a couple ways to do that. One way would be to look at all those bacterial colonies and see what proteins they're making, see if one of them is making the protein that we're looking for. We're not going to focus on that process. What we're going to look at is something called DNA or nucleic acid hybridization, and it's basically a way to say, let's look at the DNA that's in these bacteria and find which bacteria have the sequence that we're looking for, meaning the gene that we're looking for from the humans. Okay, so the way we would do this, first of all, we have to understand what hybridization is and what something called a probe is. Okay, so first of all, hybridization is so this word up here, hybridization. What that is if you take a single strand of DNA and basically attach a complementary single strand of DNA to it that's from a different source, that's called hybridization. It's making a hybrid, or something that's part, I guess, part old DNA, but part DNA from the bacteria and then parts something different, a different type of DNA. So the way you do hybridization is with something called a nucleic acid probe, and here's what we would do. If we know the gene that we're trying to study, okay, this gene of interest, then we can figure out what its sequence is, and there's, again, a long process to figure out how to do that. We're not going to focus on that process. But if we can know all or part of its DNA sequence, then we can try to search, we can make a probe to match this, so we can do hybridization. So here would be the idea. If we know, if I look at the bottom of the screen here, if I know that this is a sequence that's on the gene that I want, okay, the gene that I want to study, and it is 5' and then the sequence in 3', we can synthetically make a single strand of DNA to match that, okay? And so if I wanted to match that, what I would do, hold on, see if I can, there we go. What I would do is I would make a strand like this one that's in red here, where there's C's and G's, they match up, the A's and the T's. Everything is complementary to the gene of interest. Okay. Well, when I make a probe, what I'll do is I'll make this out of nucleotides that either have some sort of radioactive isotope in them or have some sort of like fluorescent tag on them. Okay, so that if it's radioactive, there's ways that you can identify radioactive things, and if it has a fluorescent tag, you'd be able to see it under the correct lighting situation. But basically, I'm going to take a piece of DNA that I can recognize and attach it to the gene that I want to study, and then I can actually see if that gene is present or not. But this is hybridization right here, and then this would be a DNA or a nucleic acid probe. Okay, and it matches up with a single strand. So here's our process, okay, of trying to find, again, we're trying to find which bacteria have the gene that we're actually looking for, okay. So first thing we would do is we're going to make what we call a master plate, and so we went back to that plate that was made in the previous video, and it would have bacteria in it. Some of the bacteria were blue and some of them were white. If you remember, the blue ones were bacteria we didn't want. They had picked up the plasmid, but it wasn't a recombinant plasmid. So we want the white bacteria. And we would take that white bacteria and just one by one take a couple of bacteria from the original plate. We'll say the original plate's up here. Take a couple of bacteria from a white colony up here. So let's say these are all white colonies. Okay. And we would put them in a new plate, and we're going to call this a master plate. So we're just transferring that one to here, and this one to here, and we have all these different colonies of bacteria. Transfer a couple of the cells into a new plate, a master plate, and then let them become their own colonies. Okay. So then we're taking all these white colonies, and they're in a new plate. We call this the master plate. These X's that are on here, that's we put X's on the plate in the petri dish, just for the sake of being able to know which side is up and which side's down, and it's just kind of a location. If I wanted to study this set of bacteria, I know it's the one that's closest to the two X's and whatever. So it's just to kind of give us a reference there. All right. So once we have a master plate, then what we're going to do is take a special filter paper and put it down on top of the bacterial colonies. When we do that, some of the bacteria in the colonies are going to stick onto this paper. We will also make sure that on this paper we put X's, and you can see it once we flip it off here, we put X's that match up to the X's on the master plate. That way we know where everything is. Okay, so this would pick up a little bit of bacteria from everything. All right, and then we're going to flip that over, and we're actually, this is not mentioned on this, but we're going to flip it over, and in this part right here, it's going to be a process where the bacteria that are on there, we're going to either chemically or with heat, we're going to kill those bacteria, lyse the bacteria, and denature the DNA. Okay, denaturing DNA just means it goes from being two strands together to unzipping the strands. If you denature DNA, just unzip the strands. Okay, we will also do a little chemical thing to make sure that the bacteria stick to this filter paper. Okay, so we're going to unzip the DNA in the bacteria and make sure it sticks to the filter paper. So when I flip this over, I'd have all these spots that would have different types of DNA on them that are unzipped, so they're single strand. The next thing we're going to do is take our probe, so we have a solution of our probe, we're going to pour that on top of this filter paper, and what will happen is the probes, we're going to say they're radioactive for this one instead of fluorescing, the probes are going to attach to any place that the sequence of interest, wherever that is, they'll attach to it. So they pour the probe on top of this filter paper, it attaches to anywhere where there's nucleic acid sequence that match, then they rinse off all the extra probes, and there will be some, and you can see right here, some of those probes are still attached to the genes of interest in the bacterial DNA. Well, if it's radioactive, you can take film paper and put it over top, and radioactivity, specific types of film paper, it will expose the film. If any of you have taken a traditional photography class, that makes sense to you. But it will expose the film, basically changing the color wherever the film was getting radioactivity coming off. So this film we put on top, flip the film off of there, and then we can look at it and it matches up to our original master plate. And in this one we could say none of the rest of these colonies matter, but this one, right, looks like this one right here, there, that one was glowing, and this one right here was radioactive. So those are the ones that are carrying the genes of interest that we want. Okay, so that is nucleic acid probe hybridization. That's how we can identify the bacteria that have the gene that we want. Okay. All right. Now we're gonna some extra details that are in this section here. I'm gonna do before I end this video, and then we'll move forward. Okay. This next slide here has to do with something called gene libraries. We just got done talking about getting a gene of interest out of these recombinant plasmids and identifying that. And one of the great things that scientists have done, this is kind of like sharing what they've done. I mean, you have to pay for it, but if there's been a scientist that has taken the entire human genome and chopped it up with a certain restriction enzyme and then put all of those pieces into plasmids and back into bacterial cells, and if they've gone through the work to figure out which genes are on which plasmids and all of these things, then they've done a whole lot of work, and it would have taken a lot of time. It would be a shame for every scientist in the future to have to go through that same work if they want to study a specific gene. So what scientists did pretty quickly after putting all this technology together was they came up with the idea of a gene library, and so people who have done the previous process of these recombinant plasmids and identifying what they are, they basically saved them and said, hey, if anybody wants to study the gene for human growth hormone, I've got plasmids that have that gene in them, and you can use them. Okay, and they call this a library. And so there's actually different types of libraries that are out there as far as keeping genetic information. One is the Plasmid Library, which would line up with what we had said before or shown before, where we would just have different plasmids. And what they've done is they've identified what are the genes in each of the plasmids that are inserted and what segments of the DNA, things like that. So that's the one. There's another one called a Phage Library, and you can read about it, I'm not going to get into the details of it, but essentially taking a segment of human DNA and putting it into the DNA of a virus. Okay, and they can do that in a dish. Take viral DNA, chop it open, and human DNA that's been chopped as well, and put it into a viral DNA. And then if they put it into a dish with all of the proteins for the viruses, it'll self-assemble into a new phage or into a new virus. Anyway, and then they could go through the identifying which genes were in which. But anyway, so that's a phage library. The advantage to that one, it can hold larger chunks of DNA. Bacterial plasmids are actually limited in how large of a segment of DNA they can hold. So the phages or the viruses here, they can hold larger segments. Okay. Then the other type of thing, one of the problems that we have with these gene libraries up here is that if I'm chopping human DNA, chances are wherever I'm chopping it, every now and then that's going to chop right in the middle of a gene. And if it's chopping right in the middle of a gene, then that's a gene that I can't really study. Also, say I want to work on a particular gene in this plasmid right here, there could be a whole lot of extra DNA in there, and I'm not exactly sure what DNA is being used for the actual protein that's going to be produced, so I might have some extra genes in there, or some extra whatever, and so that can be some limitations to the ones that are displayed here. So there's another option, and if I can do this here, it's called complementary DNA. You can make complementary DNA, and what they do with this complementary DNA, I'm going to make a little flow chart over here, is essentially they take messenger RNA and they can isolate messenger RNA from a cell, okay, and put it in the presence of reverse transcriptase, which you learned about when you learned about viruses in chapter 19. But reverse transcriptase is going to make a single strand of DNA, single-stranded DNA, that is complementary to the messenger RNA. Okay, and then they can basically degrade the messenger RNA so that's all that's left is a single strand of DNA. Then they can put nucleotides and enzymes in to make it a double-stranded DNA. And that double-stranded DNA is going to be the code for a specific protein. It's not going to be for just a huge chunk of DNA. It's going to be based off of the messenger RNA. All of the introns would have been cut out of the sequence already. All of the fluff would have been cut out. We know that it's the complete protein. So this can be valuable if you're looking for a very specific protein. And they've made libraries of these cDNA libraries. They're called cDNA down here. And that can be very useful as well. So these are the different gene libraries. I think it's great. The science works that way. That people, I mean, again, you have to pay for the plasmids or the phages or whatever, but the science is willing to share that stuff with each other so that you don't have to do all the work yourself. So there with that, I'm going to end this video here, and we still have more of section 1 of chapter 20 to go, but I'm going to keep putting out these short videos, and we will see you in class.